Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Diversity of Plants Zone. In this channel, you can easily find the necessary information related to plants and their diversity throughout the world. Before starting my video, I request you all to please subscribe to my YouTube channel. Today's topic of my video is about 6, 7 and 8 kingdoms of classification system. So let's start the video. First of all, I will discuss about the introduction. Kingdom in biology. In biology, a kingdom, Latin regnum, plural regna, is the second highest taxonomic rank, just below domain. Kingdoms are divided into smaller groups called phyla. Traditionally, some textbooks from United States and Canada used a system of 6 kingdoms, which are Animalia, Planty, Fungi, Protista, Archaea, which is also called as Archaebacteria, and Bacteria, which is also called as Eukaryotes. While the textbooks in Great Britain, India, Greece, Brazil, and other countries use the 5 kingdoms only, which are Animalia, Planty, Fungi, Protista, and Munera. Some recent classifications based on modern cladistics have explicitly abandoned the term kingdom, noting that the traditional kingdoms are not monophyletic, meaning that they do not consist of all the descendants of a common ancestor. The informal terms flora, which is used for plants, and fauna, which is used for animals, and in the 21st century, fungi are also used. Next, I am going to discuss about the definition and associated terms. When the Carl Linnaeus introduced the rank-based system of nomenclature into biology in 1735, the highest rank was given the name kingdom and was followed by 4 other main or principal ranks: class, order, genus, and species. Later 2 further main ranks were introduced, making the sequence kingdom, phylum, or division, class, order, family, genus, and species. In 1990, the rank of domain was introduced above kingdom. Prefixes can be added, so subkingdom, subregnum, and infrakingdom, also known as infraregnum, are the 2 ranks immediately below kingdom. Superkingdom may be considered as an equivalent of domain or empire, or as an independent rank between kingdom and domain or subdomain. In some classification systems, the additional rank branch, Latin ramus, can be inserted between subkingdom and infrakingdom, for example Protostomia and Deuterostomia, in the classification of Cavalier-Smith. So next I am going to discuss about the Cavalier-Smith's system of classification. Cavalier-Smith's system of classification, the biological classification system of life introduced by the great British zoologist Thomas Cavalier-Smith, involves systematic arrangements of all life forms on Earth, following and improving the classification systems introduced by the Carl Linnaeus, Ernst Haeckel, and Robert Whittaker, and Carl Woese. Cavalier-Smith's classification attempts to incorporate the latest developments in taxonomy. His classification has been a major foundation in the modern taxonomy, particularly with revisions and reorganizations of kingdoms and phyla. Cavalier-Smith has published extensively on the classification of protists. One of his major contributions to biology was his proposal of a new kingdom of life, the Chromista, although the usefulness of the grouping is questionable, given that it is generally agreed to be an arbitrary, polyphyletic grouping of taxa. He also proposed that all the Chromista and Alveolata shared the some common ancestor, a claim later refuted by studies of morphological and molecular evidence by the other labs. He named this new group the Chromalveolates. He also proposed and named many other high-ranked taxa like the Opisthokonta in 1987, Rhizaria in 2002, and Excavata in 2002, though he himself consistently does not include Opisthokonta as a formal taxon in his schemes. Together with the Chromalveolata Amoebozoa, he amended their description in 1998, and Archaeplastida, which he called Planty since 1981. The six formed the basis of the taxonomy of eukaryotes in the middle 2000s. He has also published prodigiously on issues such as the origin of various cellular organelles, including the nucleus and mitochondria, genome size evolution, and endosymbiosis. Though fairly well known, many of his claims have been controversial and have not gained widespread acceptance in the scientific community to date. Most recently, he has published a paper citing the paraphyly of his bacterial kingdom, the origin of Neomora from Actinobacteria, and taxonomy of prokaryotes. According to the Paleos.com, Professor Cavalier-Smith of Oxford University has produced a large body of work, which is well regarded. Still, he is controversial in a way that is a bit difficult to describe. The issue may be one of the writing style. Cavalier-Smith has tendency to make the pronouncements where others would use the declarative sentences, to use the declarative sentences where others would express an opinion, and to express opinions where angels would fear to tread. In addition, he can sound arrogant, reactionary, and even perverse. On the other hand, he has a long history of being right when everyone else was wrong. To our way of thinking, all of this is overshadowed by one incomparable virtue, the fact that he will grapple with the details. This makes for very long, very complex papers and causes all manner of dark murmuring, tearing of hair, and gnashing of teeth among those tasked with trying to explain his views of early life. See, for example, Zerzavi, 2001, and Patterson, 1999. Nevertheless, he deals with all of the relevant facts. Next, I am going to discuss about the history of 6 kingdoms of classification. 6 kingdoms. In 1977, Carl Woese and colleagues proposed the fundamental subdivision of prokaryotes into Eukaryotes, later called the bacteria, and Archaebacteria, which is later called the Archaea, based on ribosomal RNA structure. This would later lead to the proposal of 3 domains of life, which are bacteria, archaea, and eukaryota. Combined with the 5-kingdom model, this created a 6-kingdom model, where the kingdom Munera is replaced by the kingdoms bacteria and archaea. This 6-kingdom model is commonly used in the recent US high school biology textbooks, but he has received criticism for compromising the current scientific consensus. But the division of prokaryotes into 2 kingdoms remains in use with the recent 7-kingdom scheme of Thomas Cavalier-Smith, although it primarily differs in that Protista is replaced by Protozoa and Chromista. So the life is divided into the two empires. First is the empire Prokaryota, which is further divided into kingdom Eukaryotic, which is also known as bacteria. And next is the kingdom is Archaebacteria, which is also called as Archaea. And the second empire is Eukaryota, and this Eukaryota is divided into following kingdoms. First is the kingdom Protista, which is also called as Protista. Second kingdom is Planty, third kingdom is Fungi, and the fourth kingdom is Animalia, as shown into the figure. So now I am going to discuss about the six kingdoms in 1998. In 1998, Cavalier-Smith published a six-kingdom model which has been revised in subsequent papers. The variant published in 2009 is shown below. Cavalier-Smith no longer accepted the importance of the fundamental Eukaryotic, Archaebacteria divide put forward by the Woese and others and supported by the recent research. Unibacteria was divided into phyla, Archaebacteria and Posibacteria. The bimembranous unimembranous transition was thought to be far more fundamental than the long branch of genetic distance of Archaebacteria, viewed as having no particular biological significance. Cavalier-Smith doesn't accept the requirement for taxa to be monophyletic, holophyletic in his terminology to be valid. He defines Prokaryota bacteria, Negibacteria, Unibacteria and Posibacteria as the valid paraphyla. Therefore, monophyletic in the sense he uses this term taxa, making important innovations of biological significance in regard of the concept of biological niche. In the same way, his paraphyletic kingdom Protozoa includes the ancestors of Animalia, Fungi, Planty and Chromista. The advances of phylogenetic studies allowed the Cavalier-Smith to realize that the phyla thought to be archezoans and primitively amitochondriate eukaryotes had in fact secondarily lost their mitochondria, typically by transforming them into new organelles, which are also called as hydrogenosomes. This means that all living eukaryotes are in fact metakaryotes according to the significance of the term which is given by Cavalier-Smith. Some of the members of the defunct kingdom Archaezoa, like the phylum Microsporidia, were reclassified into the kingdom Fungi. Others were reclassified in kingdom Protozoa, like the Metamonada, which is now the part of infra-kingdom which is called Excavata. Because Cavalier-Smith allows paraphyly, the diagram below is an organization chart, not an ancestor chart, and doesn't represent an evolutionary tree. So the life is divided into two empires, and the first empire is Prokaryota, which is further divided into the kingdom Bacteria and includes Archaebacteria as a part of subkingdom. And the next empire is Eukaryota, and Eukaryota is further divided into the following kingdoms. First is the kingdom Protozoa, which includes Amoebozoa, Schwannozoa or Choanozoa or Excavata. Next is the kingdom Chromista, which includes Alveolata, Cryptophytes, Heteroconta like brown algae and diatoms, etc., Haptophyta and Rhizaria. Next is the kingdom Planty, which includes Glaucophytes or Glaucophytes, red and green algae and land plants. Next kingdom is the kingdom Fungi, and the last kingdom is the kingdom Animalia. Next I am going to discuss about the Six Kingdoms model. Six Kingdoms model. By 1998, Cavalier-Smith had reduced the total number of kingdoms from 8 to 6. Animalia, Protozoa, fungi, plantae, including red and green algae, Chromista, and bacteria. 5 of Cavalier-Smith's kingdoms are classified as eukaryotes as shown in the following scheme, like Eubacteria, Neomora, Archaebacteria, Eukaryotes, Kingdom Protozoa, unicorns or heterotrophs, Kingdom Animalia, and Kingdom Fungi. So biconds primarily photosynthetic. Kingdom Plantae, including red and green algae, Kingdom Chromista. Eukaryotes are divided into two major groups, which are unicont and biconds. Uniciliates are cells with only one flagellum, and uniconts are descended from the uniciliates. So unicont cells often have only one centriole as well. Biciliate cells have two flagella, and biconds are descended from biciliates, and biciliates undergo ciliary transformation by converting a younger interior flagellum into a dissimilar older posterior flagellum. Animals and fungi are unicorns, while the plants and chromists are biconds. Some protozoa are unicorns, while the others are biconds. The bacteria or prokaryotes are subdivided into Eubacteria and Archaebacteria. According to the Cavalier-Smith, Eubacteria is the oldest group of terrestrial organisms still living, and he classifies the groups which he believes are younger, Archaebacteria and Eukaryotes, as Neomora. Next I am going to discuss about the 1998 model and the Kingdom Animalia. In 1993, the Cavalier-Smith classified the Mycetozoa as a protozoan parv kingdom. By 1998, he had reclassified it as an animal subkingdom. Mycetozoa contains 3 phyla, which are Mycetosporidia, Haplosporidia, and Paramixia, which were reclassified as animals along with Mycetozoa. Likewise, Cavalier-Smith reclassified the protozoan phylum Mesozoa as an animal subkingdom. In his 1998 scheme, the animal kingdom was divided into 4 subkingdoms, which are Radiata, Phyla, Porifera, Cnidaria, Placozoa, and Ctenophora. And next is the Myxozoa and Mesozoa and Bilateria, all other animal phyla. He created the 5 new animal phyla, which are Acanthognatha, which includes rotifers, Acanthocephalans, Gastrotrix, and Gnathostomulates. Next is the Brachiozoa, which includes brachiopods and phoronids. Next is the Lobopoda, which includes onychophorans or onychophorans. And the next is the tardigrades. Next is Camptozoa, which includes Entoprocta and Symbion. And next is the Nemathalanthes, which includes Nematoda, Nematomorpha, Loricifera, and Priapolida. And the next is the Chinorhyncha, and recognize the total of 23 animal phyla as shown here. Animal Kingdom Animalia, Subkingdom Radiata, Infrakingdom Spongiaria, Phylum Porifera, Infrakingdom Coelenterata or Coelenterata. Next phylum is Cnidaria, and the next phylum is the Ctenophora. infrakingdom Plecozoa, and the subkingdom is the Mycetozoa. Phylum Myxosporidia, subkingdom Bilateria, branch Protostomia, infrakingdom Lophozoa, superphylum Polyzoa, phylum Bryozoa, phylum Camptozoa, Entoprocta, and Cyclophora. And next is the superphylum. Superphylum is the Conchozoa, and the phylum Mollusca, phylum Brachiozoa, Sensulatu, and Branchiozoa and Phoronida, superphylum. And the next superphylum is the Siponcula, and the phylum is the Siponcula, superphylum Vermizoa, and the phylum is the Nemertina. Next phylum is Annelida. Infrakingdom is the Chaetognathae or Chaetognathae. Phylum Chaetognathae or Chaetognathae. Phylum is the Chaetognatha or Chaetognatha. Infrakingdom is the Ichthyozoa, superphylum Nemathelminthes, phylum Nemathelminthes, Nematoda and Nematomorpha, and Parapozoa. Next is the Chinorhyncha and Loricifera are included in it. Superphylum Hemopoda, phylum Lobopodia, which includes Onychophora and Tardigrada. Phylum Arthropoda. Infrakingdom is the Platyzoa. Phylum is the Platyhelminthes, which includes Xenicoelomorpha. Phylum Acanthognatha, which includes Rotifera and Acanthocephala. And next is the Gnathostomolids or Gnathostomolida. Next is the Gastrotricha. Next is the branch Deuterostomia. Infrakingdom is the Coelomorpha or Coelomopora. Phylum Echinodermata. Phylum Hemicodata. Infrakingdom Cordonia. Phylum Urocorda. Phylum Chordata. Next is the subkingdom, which is Mesozoa. Phylum is the Mycetozoa. Next I am going to discuss the kingdom Protozoa. Under Cavalier-Smith proposed classification system, Protozoa share the following traits. They have or are descended from the organisms with the mitochondria. They have or are descended from organisms with the peroxisomes. They lack the collagenous connective tissue. They lack the epicellary retronemes, rigid thrust reversing, tubular ciliary hairs, and they lack the two additional membranes outside their chloroplast envelope. Organisms that do not meet these criteria were reassigned to the other kingdoms by Cavalier-Smith. So next I am going to discuss about the 2003 model, which includes the kingdom Protozoa. In 1993, Cavalier-Smith divided the kingdom Protozoa into 2 subkingdoms and 18 phyla. By 2003, he used the phylogenetic evidence to revise the total number of proposed phyla down to 11: Amoebozoa, Choanozoa, Cercozoa, Reiteria, Leucozoa, Metamonada, Euglenozoa, Percolozoa, Apusozoa, Alveolata, Celiophora, and Meiozoa. Next is the unicorns and bicorns. Amoebozoa do not have flagella and are difficult to classify as unicant or bicants based on their morphology. In his 1993 classification scheme, Cavalier-Smith incorrectly classified amoebas as bicants. Gene-fusion research later revealed that the clade Amoebozoa was ancestrally uniciliate. In his 2003 classification scheme, Cavalier-Smith reassigned Amoebozoa to the unicant clade, along with animals, fungi, and the protozoan phylum Choanozoa. Plants and all other protists were assigned to the clade bicant by the Cavalier-Smith. Cavalier-Smith's 2003 classification scheme. Unicants, protozoan phylum Amoebozoa ancestrally uniciliate. Opisthocorns, uniciliate protozoan phylum Choanozoa, kingdom Fungi and kingdom Animalia. Bicants, protozoan infrakingdom Rhizaria, phylum Cercozoa, phylum Deuteria, which includes Radiozoa and Foraminifera. Protozoan infrakingdom Excavita, which includes the phylum Leucozoa and the phylum Metamonada. Phylum Euglenozoa, phylum Percolozoa, protozoan phylum Apusozoa. Ticomonadia and Diphylida, the chromalveolate clade, kingdom Chromista, which includes Cryptista and Heteroconta and Haptophyta. Protozoan infrakingdom Alveolata, phylum Seriophora, phylum Meiozoa, which includes Protalveolata, Dinozoa, and Apicomplexa. Kingdom Plantae includes Viridiplantae, Rhodophyta, and Glaucophyta or Glucophyta. So next I am going to discuss about the Cladogram of Life. By the September 2003, Cavalier-Smith's tree of life looked like this. In the tree below, the traditional plant and animal fungal kingdoms, as well as Cavalier-Smith's proposed kingdom Chromista, are shown as leaves. The leaves Eukaryotic and Archaebacteria together make up the kingdom Bacteria. All remaining leaves together make up the kingdom Protozoa. So, Eukaryotic and Neomora are further divided into Archaebacteria and Eukarya. Eukarya is further divided into bicants and unicants, and bicants are further divided into the Apusozoa and Cubozoa. Cubozoa is further divided into the Excavita and Rhizaria. On the other hand, Rhizaria is divided into Reteria and Cercozoa. Apusozoa is further divided into the two kingdoms, which are the kingdom Plantae and Chromalveolata. Chromalveolata is further divided into the kingdom Chromista and Alveolata. On the other hand, unicants are further divided into Amoebozoa and Opisthocorns. And Opisthocorns are further divided into the kingdoms like the Choanozoa and kingdom Animalia. And the next kingdom is the kingdom Fungi, as shown into the figure. By 2006, Cavalier-Smith's microbial tree looked like this. By 2010, new data emerged that showed that unicants and bicants originally considered to be separate because of an apparently different organization of cilia and cytoskeleton are in reality more similar than previously thought. As a consequence, Cavalier-Smith revised the tree and proposed to move its root to reside in between the Excavita and Euglenozoa kingdoms. So, This is our Cavalier-Smith's Trees of Life as shown into the figure 2006. So first is the Chlorobacteria. A stands for the gram-negative with the peptidoglycan cell wall like the chlorosome, Chlorobacteria. B. Oxygenate photosynthesis, OMP85 and 4 new catalases, Haeckel. C. Glycobacterial revolution, outer membrane with insertion of lipopolysaccharides, hopanoids, and diaminopimalic acid, Tol C and Ton B. Next is D. Phycobilin chromophores and cyanobacteria. E. Flagella. And F. 4 sections, an amino acid in HSP60 and FTSC, and a domain in RNA polymerases beta and alpha. Gracilicuetes. Gracilicuetes is further divided into the Spirochaetae and further divided into the Sphingobacteria and Planktobacteria PVC and Proteobacteria. So next is the G. G stands for the endospores, and the G after that is H. Gram-positive bacteria. Hypertrophy of the wall, peptidoglycan sortase enzyme, and a loss of the outer membrane. I is used for the glycerol 1,P dehydrogenase. And next is Eurybacteria. So Eurybacteria is further divided into the Endobacteria, which is also called Firmicutes. Next is the J. Proteasome and phosphatidyl inositol. So J is further divided into the K. K shows neomolar revolution and replacement of the peptidoglycan by the glycoproteins and lipoproteins, and divided into the actinobacteria. So K is further divided into the L. L stands for the reverse DNA gyrase and the ether lipid isoprenoids, Archaea. And next is the M. M stands for the phagocytosis and it forms Eukarya, as shown into the figure. So this is the Cavalier-Smith's Tree of Life 2006 as shown into the figure. Next I am going to discuss about the history of 7 kingdoms of classification. 7 kingdoms. Cavalier-Smith and his collaborators revised their classification in 2015. In this scheme, they reintroduced the division of prokaryotes into 2 kingdoms, which is bacteria. Bacteria is also called as Eukaryotic, and Archaea, which is also known as Archaebacteria. This is based on the consensus in the taxonomic outline of bacteria and Archaea, TOBA, and the Catalogue of Life. So the life is divided into 2 empires. First empire is the Prokaryota, which is further divided into kingdom bacteria, and the next kingdom is Archaea. Second empire is the Eukaryota. So the Eukaryota is divided into the kingdom Protozoa, for example, Amoebozoa, Choanozoa, and Excavita. Second kingdom is the Chromista, for example, Alveolata, Cryptophytes, Heteroconta, which includes brown algae and diatoms, etc., Haptophyta, and Rhizaria. Third kingdom is Planty, for example, Glaucophytes or Glucophytes, red and green algae, and land plants. Fourth kingdom is Fungi, and the fifth is kingdom Animalia. Next I am going to discuss about the Seven Kingdoms model. Seven Kingdoms model. In 1987, Cavalier-Smith introduced a classification divided into two superkingdoms, Prokaryota and Eukaryota, and the seven kingdoms, two prokaryotic kingdoms which are Eubacteria and Archaebacteria, and five eukaryotic kingdoms which includes Protozoa, Chromista, Fungi, Planty and Animalia. Cavalier-Smith and his collaborators revised the classification in 2015 and published it in PLOS 1. In this scheme, they reintroduced the classification with the division of prokaryotes superkingdom into two kingdoms which are bacteria, also known as Eubacteria, and Archaea, which is also called as Archaebacteria. This is based on the consensus in the taxonomic outline of bacteria and Archaea, TOBA, and the Catalogue of Life. The Seven Kingdoms of Life. Introduction. The seven kingdoms of living things are divided into two major groups, which are prokaryotes and eukaryotes. There are two prokaryotic kingdoms and five eukaryotic kingdoms. There are huge fundamental differences between the ways these two groups go about living. First, I am going to discuss about the eukaryotes. So eukaryotes have a separate membrane-bound nucleus, numerous mitochondria and organelles. These areas are separated from the main mass of the cell's cytoplasm by their own membrane, in order to allow them to be more specialized in their activity. Organelles include things such as Golgi apparatus body, mitochondria, ribosomes, lysosome, and vesicles, and of course the nucleus. So this is a eukaryote cell with clear nucleus, red, and ovoid-shaped organelles as shown into the figure. The nucleus contains all the eukaryote cell DNA, for instance, and mitochondria are where energy is generated. The exception to this rule are red blood cells, which have no nucleus and do not live very long. So next, I am going to discuss about the prokaryotes. Prokaryotes do not have a nucleus, mitochondria, or any other membrane-bound organelles. In other words, neither their DNA nor any other of their metabolic functions are collected together in a discrete membrane-enclosed area. Instead, everything is openly accessible within a prokaryotic cell. So this is a prokaryote cell with no nucleus or organelles as shown into the figure. Though some bacteria have internal membranes as sites of metabolic activity, these membranes do not enclose a separate area of the cytoplasm. There are also many other differences between the prokaryotic and eukaryotic cells. So next I am going to discuss about eukaryotic kingdoms and the prokaryotic kingdoms. The next level down of classification is into kingdoms. Older books will teach that there are two kingdoms, plants and animals. Books produced towards the end of the last century will generally list 6 kingdoms as of 2015, and the publication of a higher level classification of all living organisms by the Ruggiero et al. 7 kingdoms are now recognized. The changes have resulted from better information collected from the scanning, electron microscope imaging, and DNA analysis. However, even this pattern of organization is not finished, and there may well be changes in the future. Unfortunately, botanists and mycologists and zoologists who study the three kingdoms you will be most concerned with do not always use the same terminology below kingdom level, though much of it is the same. Here you will find a brief description of the 7 kingdoms of life. This will hopefully link in time to more information, including more terminology. So next I am going to discuss further about the 7 kingdoms of life table. So in this table, first I will discuss about kingdom, when evolved, their structure and photosynthesis. So first kingdom is the prokaryotic kingdoms, which include bacteria. Bacteria is evolved about 3 to 4 billion years ago. They are unicellular and photosynthesis sometimes. Next is the Archaea. Archaea evolved 3 to 4 billion years ago. In their structure, they are unicellular and photosynthesis no. Second kingdom is the eukaryotic kingdoms, which includes Protozoa. Protozoa evolved about 1.5 billion years ago. In their structure, they are unicellular and photosynthesis doesn't take place. Next is the Chromista, which has evolved about 1.2 billion years ago, and in their structure, they are unicellular. Photosynthesis originally but sometimes lost chlorophyll C. So the next kingdom is the fungi, which has evolved about 1 billion years ago. In this structure, they may be unicellular or multicellular. Photosynthesis doesn't take place. Next is Animalia, which has evolved about 700 million years ago. In this structure, they are multicellular and photosynthesis doesn't take place. Next is the kingdom Planty, which is evolved about 500 million years ago, and their structure is multicellular. Photosynthesis, yes, takes place because chlorophyll is present in them, and mostly chlorophyll is chlorophyll C. The Chromista and 3 multicellular eukaryotic kingdoms all evolved. As best we understand from the Protozoa, the 2 kingdoms of unicellular eukaryotic organisms are considered to be polyphyletic, meaning that they do not necessarily represent a single coherent clade taxonomically. While the 3 multicellular kingdoms are all quite distinct, they share some characteristics and not others. Overall, most scientists these days consider the fungi to be more closely related to animals than plants. The table below highlights the major differences between the 3 kingdoms but doesn't most characteristics that are shared by all the 3 to some extent, such as having some members that can reproduce asexually. So next I am going to discuss about the 3 multicellular kingdoms. First I will discuss their characteristic, then kingdom Planty, then kingdom Animalia, and then kingdom Fungi. So first characteristic is cell wall. Kingdom Planty, cellulose. Kingdom Animalia, absent. Kingdom Fungi, chitin. Next characteristic is reproduction. Kingdom Planty, gametes or spores. Kingdom Animalia, gametes. Kingdom Fungi, spores. So next characteristic is mitosis. Kingdom Planty, open. Kingdom Animalia, open. Kingdom Fungi, open or closed. Next characteristic is cellular differentiation. Kingdom Planty, in whole organism. Kingdom Animalia, in whole organism. Kingdom Fungi, only for fruiting bodies. Next characteristic is the haploid cells versus diploid cells. Kingdom Planty, most body cells are diploid. Kingdom Animalia, most body cells diploid. Kingdom Fungi, most body cells haploid. And the next character is the food acquisition. Kingdom Planty, autotrophic. Kingdom Animalia, heterotrophic. Kingdom Fungi, heterotrophic. And the next characteristic is the chlorophyll presence. Kingdom Plantae, present. Kingdom Animalia, absent. Kingdom Fungi, absent. And the next character is food storage. Kingdom Plantae, starch. Kingdom Animalia, glycogen. Kingdom Fungi, glycogen. And the next character is the motility. The whole organism can move itself or not. So Kingdom Plantae, no. Kingdom Animalia, mostly yes. Kingdom Fungi, no. Next I am going to discuss about the 8 kingdoms model. 8 kingdoms system, Cavalier-Smith's concept. The kingdom Protista was still too diverse to be taxonomically useful. Many attempts have been made to divide the protists into better defined kingdoms, and in this regard, an 8 kingdom system was given by Cavalier-Smith 1987. Cavalier-Smith, using ultrastructural characteristics as well as ribosomal RNA sequences, divides all organisms into 2 empires and 8 kingdoms. The Archaebacteria consist of 2 kingdoms, the Eukaryotic and Archaebacteria. The empire Eukaryota contains 6 kingdoms of eukaryotes, which are Archaezoa, Protozoa, Chromista, Fungi, Planty and Animalia. Archaezoa and Chromista are the 2 new kingdoms of eukaryotes. The kingdom Archaezoa consists of primitive eukaryotic unicellular microorganisms, for example, Giardia or Giardia, that possesses 70 subunit ribosomes and lactocell organelles like Golgi apparatus, mitochondria, chloroplast and peroxisomes. The kingdom Chromista is represented by diatoms, brown algae, cryptomonads, and oomycetes, mainly the photosynthetic microorganisms that have their chloroplasts within the lumen of the rough endoplasmic reticulum, rather than in the cytoplasmic matrix, as is found in the members of kingdom Plantae. The 4 kingdoms like Plantae, Fungi, Animalia, and Protozoa are retained, but their boundaries have been adjusted to better define each kingdom and differentiate them from the others. So, this is the 8 kingdom system according to Cavalier-Smith 1987. So, the empire Eukaryota includes Plantae, Chromista, Fungi, Animalia, Protozoa, and Archaezoa. On the other hand, the Archaebacteria includes Archaebacteria and Eukaryotes as shown into the figure. So next, I am going to discuss further about the 8 kingdoms model. The first 2 kingdoms of life are Plantae and Animalia. The use of word kingdom to describe the living world dates as far back as Linnaeus. The time period was 1707 to 1778, who divided the natural world into 3 kingdoms, which are animal, vegetable, and the minerals. The classifications, animal kingdom or kingdom Animalia and plant kingdom or the kingdom Plantae, remain in use by modern evolutionary biologists. The Protozoa were originally classified as members of the animal kingdom. Now they are classified as multiple separate groups. The third kingdom, Protista. By mid-19th century, the microscopic organisms were generally classified into 4 groups, which are Protozoa, which includes primitive animals, Protophyta, which includes the primitive plants, and Phytozoa, animal-like plants or plant-like animals. And in 1858, Richard Owen, the time period is 1804 to 1892, proposed that the animal phylum Protozoa be elevated to the status of kingdom. In 1860, John Hogg, the time period was 1800 to 1869, proposed that the Protozoa and Protophyta be grouped together into a new kingdom, which he called Regnum Primigenum. Kingdom primitive. According to Haeckel, this new classification scheme prevented the unnecessary trouble of conducting about their supposed natures and of uselessly trying to distinguish the Protozoa from the Protophyta. In 1866, the Ernst Haeckel, in 1834 to 1919, proposed the name of the Protista for the primigenial kingdom and included the bacteria in this third kingdom of life. So, this is the sea anemone is an animal that resembles a plant as shown into the figure. Next I am going to discuss further about the 8 kingdoms model. The 4th kingdom is fungi. By 1959, Robert Whittaker proposed that the fungi which were formerly classified as plants be given their own kingdom. Therefore, he divided the life into 4 kingdoms such as the Protista, which includes the unicellular organisms. Next is Planty, which includes the multicellular plants, fungi, and Animalia or multicellular animals. So Whittaker subdivided the Protista into 2 subkingdoms, which are Monera, Bacteria, and Eunucleata, which includes the single-celled eukaryotes. So this is a Japanese popular mushroom, clockwise from left, and this is the Enokiteki, and this is the Benashimiji, and this is the Benapi shimiji, and this is king oyster mushroom, and the next is the shiitake as shown into the figure. The 8 kingdoms model, the 5th kingdom is bacteria, which is also called as Monera. So bacteria are fundamentally different from the eukaryotes, which are plants, animals, fungi, amoebas, protozoa, and Chromista. So eukaryotes have cell nuclei, bacteria do not. In 1969, the Whittaker elevated the bacteria to the status of kingdom, and his new classification system divided the living world into 5 kingdoms. These 5 kingdoms are Planty, Animalia, Protista or Eunucleata, and fungi and Monera, the kingdom of bacteria. The 6th kingdom is Archaebacteria. The kingdom Monera can be divided into 2 distinct groups, which are Eukaryotic or also called as true bacteria, and Archaebacteria, which is also called as Archaea. In 1977, the Carl Woese and the George E. Fox established that Archaebacteria are methanogens. In their case, were genetically different based on their ribosomal RNA genes from bacteria, so that life could be divided into 3 principal lineages, namely Eubacteria, all typical bacteria, Archaebacteria or methanogens, and Eukaryotes include all eukaryotes. In 1990, was introduced the domain above kingdom by creating the 3 domain system, such as bacteria, archaea, and eukarya. But Cavalier-Smith considered Archaebacteria as a kingdom. So this is a phylogenetic tree which is based on the Woese ribosomal RNA analysis. So first kingdom is bacteria. So bacteria includes Chloroflaxi, Gram-positives, Spirochetes, Proteobacteria, Cyanobacteria, and Planktomyces, Bacteroides, Cytophaga, Thermotoga, and Equifax. On the other hand, Archaea includes Haloarchaea, Methanosarcina, Methanobacterium, Methanococcus, Thermococcus, Celer, Thermoproteus, Pyrodicticum. And the next is Eukaryota, which includes ant-amoebae, slime molds, animals, fungi, plants, ciliates, flagellates, trichomonads, microsporidia, and diplomonads. So the seventh kingdom is the Chromista. By 1981, Cavalier-Smith had divided all the eukaryotes into 9 kingdoms. In it, he created the Chromista for a separate kingdom of some protists. Most chromists are photosynthetic, and this distinguishes them from most of other protists which lack the photosynthesis. In both plants and chromists, photosynthesis takes place in the chloroplasts. In plants, however, the chloroplasts are located in the cytosol, while in chromists, the chloroplasts are located in the numen of their rough endoplasmic reticulum. This distinguishes the chromists from plants. Based on the addition of Chromista as a kingdom, he suggested that even with his 9 kingdoms of eukaryotes, the best one for general scientific use is a system of 7 kingdoms, which includes Planty, Animalia, Protozoa, Chromista, Fungi, Eubacteria, and Archaebacteria. So next is the 8th kingdom, which is Archaezoa. In 1983, the Cavalier-Smith introduced Archaezoa, for which he called the primitive protists that lacked the mitochondria. He originally considered it as a subkingdom, but by 1989, with the establishment of Chromista as a separate kingdom, he treated it as a kingdom. Archaezoa is now defunct, and he now assigns former members of the kingdom Archaezoa to the phylum Amoebozoa. So next I am going to discuss about the kingdom Protozoa Sensulata Cavalier-Smith. So Cavalier-Smith referred to what remained of the protist kingdom after he removed the kingdoms like Archaezoa and Chromista as the kingdom Protozoa. In 1993, this kingdom contained 18 phyla as summarized in the following table. So, in this table, first I will discuss phylum, then assigned to, then characteristic, and then fate. So, the first is the Parcolozoa, assigned to the subkingdom Dictyozoa. Characteristic lacks Golgi dictyosomes. Next is the Paramixia, which is assigned to the subkingdom Dictyozoa branch Paramixia. Characteristic has Golgi dictyosomes, lacks mitochondria. And the third is Euglenozoa, which is assigned to the subkingdom Dictyozoa branch Biconta, infrakingdom Euglenozoa, and characteristic has Golgi dictyosomes and mostly with mitochondria with transplicing of the mini axons. Fourth is the Opalozoa, which is also known as the flagellates, and this is assigned to the subkingdom Dictyozoa branch Biconta, infrakingdom Neozoa, parvkingdom Coeliomixa, and superphylum is Opalomixa. Characteristic has Golgi dictyosomes, tubular mitochondrial cristae with the cis-spliced introns, predominantly ciliated, no cortical alveoli. And fifth is the Mycetozoa, which is also known as the slime molds, and this is assigned to the subkingdom Dictyozoa branch Biconta, infrakingdom Neozoa, parvkingdom Coeliomixa, superphylum Opalomixa. Characteristic has Golgi dictyosome, tubular mitochondrial cristae with the cis-spliced introns and predominantly ciliated, no cortical alveoli. And the sixth is the Choanozoa or choanoflagellates, and this is assigned to the subkingdom Dictyozoa branch Biconta, infrakingdom Neozoa, parvkingdom Coeliomixa, superphylum Choanozoa, and the characteristic has Golgi dictyosomes, flattened mitochondrial cristae with the cis-spliced introns and predominantly ciliated, no cortical alveoli. Seventh is the Dinozoa and Dinoflagellata and the Protalveolata. This is assigned to the subkingdom Dictyozoa branch Biconta, infrakingdom Neozoa, parvkingdom Alveolata, superphylum is Meozoa. Characteristic has Golgi dictyosomes, tubular mitochondrial cristae with the cis-spliced introns with cortical alveoli. Fate reassigned to Meozoa in Alveolata. So eighth is the Apicomplexa. Apicomplexa assigned to the subkingdom Dictyozoa branch Biconta, infrakingdom Neozoa, parvkingdom Alveolata, superphylum Meozoa, Characteristic has Golgi dictyosomes, tubular mitochondrial cristae with the cis-spliced introns with cortical alveoli, and fate reassigned to Mesozoa in alveolata. Ninth is the Coeliophora, which is assigned to the subkingdom Dictyozoa, branch by conta, infrakingdom Neozoa, parvkingdom Alveolata, superphylum Heterokaryota. Characteristic has Golgi dictyosome, tubular mitochondrial cristae with the cis-spliced introns with cortical alveoli. Fate reassigned to alveolata. So 10th is the Rhizopoda, Lobos and Phylos amoebae, assigned to the subkingdom Dictyozoa, branch biconta, infrakingdom Neozoa, parvkingdom Neosarcodina. So next is the characteristic has Golgi dictyosomes usually with the tubular cristae with the cis-spliced introns. 11th is the Reticulosa, and this is the Foraminifera reticulopodial amoebae, which is assigned to the subkingdom Dictyozoa, branch biconta, infrakingdom Neozoa, parvkingdom Neosarcodina, and the characteristic has Golgi dictyosomes usually with the tubular cristae with the cis-spliced introns. 12th is the Heliozoa. Subkingdom is the Dictyozoa, branch biconta, infrakingdom Neozoa, parvkingdom Actinopoda. Characteristic has Golgi dictyosomes and mostly with the mitochondria with the cis-spliced introns has exopodia. So 13th is the Radiozoa, assigned to subkingdom Dictyozoa, branch biconta, infrakingdom Neozoa, parvkingdom Actinopoda. Characteristic has Golgi dictyosomes mostly with the mitochondria with the cis-spliced introns has exopodia. So 14th is the Antamebia. So this is assigned to the subkingdom Dictyozoa, branch biconta, infrakingdom Neozoa, parvkingdom Antamebia. So characteristic has Golgi dictyosomes with the cis-spliced introns, no mitochondria, peroxisomes, hydrogenosomes or cilia, which is transient intranuclear centrosomes. And the 15th is the Myxosporidia, which is assigned to the subkingdom Dictyozoa, branch biconta, infrakingdom Neozoa, parvkingdom Myxozoa. Characteristic has Golgi dictyosomes with the cis-spliced introns, endoparasitic multicellular spores, mitochondria and no cilia. So this is reclassified as animals in 1998. 16th is the Haplosporidia. This is assigned to the subkingdom Dictyozoa, branch biconta, infrakingdom Neozoa, parvkingdom Myxozoa. Characteristic has Golgi dictyosomes with the cis-spliced introns, endoparasitic multicellular spores, mitochondria and no cilia. So fate reclassified as animals in 1998. 17th is the Paramixia. This is assigned to the subkingdom Dictyozoa, branch biconta, infrakingdom is Neozoa, parvkingdom Myxozoa. Characteristic has the Golgi dictyosomes with the cis-spliced introns, endoparasitic multicellular spores, mitochondria and no cilia. So fate reclassified as animals in 1998. So 18th is the Mesozoa or Myxozoa. So this is assigned to the subkingdom Dictyozoa, branch biconta, infrakingdom Neozoa, parvkingdom, and this is the Myxozoa. Characteristic has Golgi dictyosomes with the cis-spliced introns, tubular mitochondrial cristae with the multicellular with no collagenous connective tissue. And fate, this is reclassified as animals in 1998. So next I am going to discuss about the summary of the kingdom systems of classification. So Linnaeus system was first introduced in 1735 and he introduced two kingdoms which are Vegetalia and Animalia. Next is the Haeckel. In 1866 he introduced three kingdoms which are Protista, Planty and Animalia. Next is Chatham. In 1925 he introduced two empires which are Prokaryota and Eukaryota. Fourth is Copeland. In 1938 he introduced the two empires and four kingdoms which are Monera, Protista, Planty and Animalia. Next is Whittaker. In 1969, he introduced 2 empires and 5 kingdoms, which are Munera, Protista, Planty, Fungi, and Animalia. Next is Woese et al. In 1977, he introduced 2 empires and 6 kingdoms, which are Eukaryotic, Archaebacteria, Protista, Planty, Fungi, and Animalia. Next is Woese et al. Again. In 1990, he introduced 3 domains, which are Bacteria, Archaea, and Eukarya. Next is Cavalier-Smith. In 1993, he introduced 3 superkingdoms and 8 kingdoms, which are Eukaryotic, Archaebacteria, Archaezoa, Protozoa, Chromista, Planty, Fungi, and Animalia. So next is Cavalier-Smith. In 1998, he introduced 2 empires and 6 kingdoms, which are Bacteria, Protozoa, Chromista, Planty, Fungi, and Animalia. And at last is Ruggiero et al. In 2015, he introduced 2 superkingdoms and 7 kingdoms, which are Bacteria, Archaea, Protozoa, Chromista, Planty, Fungi, and Animalia, as shown into the figure. So, the kingdom level classification of life is still widely employed as a useful way of grouping the organisms, notwithstanding some problems with this approach. So, kingdoms such as Protozoa represent grades rather than clades, so are rejected by the phylogenetic classification systems. The most recent research doesn't support the classification of eukaryotes into any of the standard systems. As of April 2010, no set of kingdoms is sufficiently supported by research to attain the widespread acceptance. So, in 2009, Andrew Roger and Alastair Simpson emphasized the need for the diligence in analyzing the new discoveries with the current pace of change in our understanding of the eukaryote of life. So we should proceed with the question. Next, I am going to discuss about the eukaryotic subgroups. In 2004, a review article by the Simpson and Roger noted that Protista were a grab bag for all the eukaryotes that are not animal, plants, or fungi. They held that only monophyletic groups should be accepted as formal ranks in a classification, and that while this approach had been impractical previously, necessitating literally dozens of eukaryotic kingdoms, and it had now become possible to divide the eukaryotes into just a few major groups that are probably all monophyletic. On this basis, the diagram opposite redrawn from their article showed the real kingdoms, their quotation marks of the eukaryotes. A classification which followed this approach was produced in 2005 for the International Society of Protologists by the committee which worked in the collaboration with the specialists from many societies. It divided the eukaryotes into same 6 supergroups. The published classification deliberately didn't use formal taxonomic ranks, including that of kingdom. So the life is divided into domain Bacteria, which is further divided into Bacteria. Next domain is Archaea, which is further divided into Archaea. And the third domain is Eukaryota, which is divided into Excavita. Various flagellate protozoa are included in it. Next is Amoebozoa, which includes the most lobose amoeboids and slime molds. Next is the Opisthoconta, which includes the animals, fungi, quinoflagellates, etc. And next is Rhizaria, which includes the 4 Aminifera, Radiolaria, and various other amoeboid protozoa. And the next is the Chromalvulata, which includes the Stramanopeles like brown algae and diatoms, etc. and Haptophyta, Cryptophyta or Cryptomonads and Alveolata. And next is the Archaeplastida or Primoplanti, like the land plants, green algae, red algae, and glaucophytes. So this is the phylogenetic and the symbiogenetic tree of the living organisms, showing the origins of the eukaryotes and prokaryotes. So eukaryota includes the Planty, Protista, Fungi, and Animalia. So on the other hand, the prokaryota includes the Bacteria and Archaea, as shown into the figure. So Next I'm going to discuss further about the eukaryotic subgroups in this system. The multicellular animals like the Mycetozoa are descended from the same ancestor as both the unicellular choanoflagellates and the fungi which form the Opisthokonta. So plants are thought to be more distinctly related to the animals and fungi, and however in the same year as the International Society of the Protistologists classification was published in 2005, and doubts were being expressed as to further whether some of these supergroups were monophyletic, particularly the Chromalveolata, and review in the 2006 noted the lack of evidence for several of the 6 proposed supergroups. As of 2010, there is widespread agreement that the Rhizaria belong with the Stramanopeles and the Alveolata in a clade dubbed the SAR subgroup. So that the Rhizaria is not one of the main eukaryote groups. Beyond this, there doesn't appear to be a consensus. And the Rogozin et al. In 2009 noted that the deep phylogeny of eukaryotes is an extremely difficult and controversial problem. As of the December 2010, there appears to be a consensus that the 6 supergroup model proposed in 2005 doesn't reflect the true phylogeny of the eukaryotes and hence how they should be classified, although there is no agreement as to the model which should replace it. So this is one of the hypotheses of the eukaryotic relationship which is depicted by the Alastair Simpson. So first is the Archaeplastida. Archaeplastida is a supergroup which includes the glaucophytes, red algae, green algae plus plants. Next is the Amoebozoa, which includes the Lobosa amoebae, Variosea, and Mycetozoa, and Archaeamoebae are included in it. Next supergroup is the Opisthokonta. So this Opisthokonta includes the animals, choanoflagellates, and Philasteria, and the next is the Ichthyosporia, and fungi and the nuclearides, and the next is the supergroup of the CRUMS, and CRUMS are included in it, and it includes the Eposomonads, Breviates, and the Collodictyonids, and Rigiphyllates, and next is the Mentamonas. And the next is the supergroup of the SAR, and the SAR includes, and SAR is a supergroup which contains the Straminopeles and alveolates, and Rhizaria are included in it. Next supergroup is the Haptista. So Haptista includes the Rapimonads, Haptophytes, and Centrohylids are included in it. Next supergroup is the Cryptista. So Cryptista includes the Cryptomonads and Catabel ferrets. Catabel ferrets are included in it, and Palpitomonas are included in it. And next is the Picozoa. And next supergroups is the Hemimestigophora, and next is the Discoba. So Discoba includes the Glenozoa and Heterolobosea, and Metamonata and Malavimonads, and excavate, and these are all included into the excavates. And next is the Ensiromonads. So these are all belong to the eukaryotes, and this is one of the hypotheses of eukaryotic relationships which is depicted by the Alastair Simpson as shown into the figure. So next I am going to discuss about the viruses. So viruses, the International Committee on Taxonomy of Viruses uses the taxonomic rank kingdom for the classification of viruses with the suffix "vari", but this is beneath the top level classifications of realm and the subrealm. There is ongoing debate as to whether the viruses can be included in the tree of life. The 10 arguments against include the fact that they are obligate intracellular parasites that lack the metabolism and are not capable of replication outside a host cell. Another argument is that their placement in the tree would be problematic, and since it is suspected that viruses have arisen multiple times and they have the penchant for the harvesting nucleotide sequences from their hosts, and on the other hand, arguments favor their inclusion. So one comes from the discovery of unusually large and complex viruses, such as the mimivirus that possesses the typical cellular genes. So this is a table. So first I will discuss the taxonomic root node. Next, the 2 superdomains controversial, and 2 empires, 3 domains, 5 dominiums. Next is the 5 kingdoms, 6 kingdoms, and eocide hypothesis. So taxonomical root node includes the biota, vitae, or life. So 2 superdomains are acytota or aphanobionta, non-cellular life, or cytota, which shows the cellular life. 2 empires are prokaryota, prokarya or monera, eukaryota or eukarya, and the 3 domains are bacteria, archaea or eukarya. So 5 dominiums are the virusobiota, viruses or viroids. So next is the parinobiota or parinoids or parians, and next is the bacteria or archaea. The 5 kingdoms are monera, protista, fungi, planty and animalia. The 6 kingdoms are Eubacteria, Archaebacteria, Protista, fungi, planty and animalia. So eocide hypothesis includes the bacteria, archaea, including eukaryotes. So, if you like my video, please subscribe to my YouTube channel and press the bell icon as well for further notifications. Thanks for watching. Thank you so much.